Okay, so good morning and good evening, everyone. So welcome to today's ImmuZoom seminar. Today, it's my great pleasure to welcome today's speaker, Dr. Zhong from Shanghai University. So currently, Yi is a principal investigator in Shanghai University School of Medicine. His lab is based in the Renji Hospital in Shanghai. A very brief introduction about Yi. So Yi actually completed his undergraduate study in University of South China, and then continued to pursue his master's degree in Shanghai University in the Department of Microbial Genomics. Afterwards, he had a very brief training in University of Tübingen with Max Planck Institute in Germany from 2011 to 2012. So from 2012, he actually started his graduate study in computational biology, also in Tübingen, and then together with Memorial Sloan Kettering Cancer Center. So after he complete his PhD training, he continued to pursue his postdoc training with Sasha Rudensky in Sloan Kettering immunology program in New York from 2016 to 2021. And then from 2021, he actually started his independent research program, as I just mentioned, in Shanghai University School of Medicine. So currently, Yi's lab is mainly focused on several different points, particularly with T-cell genetics, and cover different aspects, including system immunology, epigenomics, and gene regulation, and also computational biology and statistical modeling, and then also the mRNA translational regulation, I think on the cover of or on the general arch of cancer immunology. So I think today he's gonna talk about a little bit of his past work, maybe a bit of the current work related to the epigenetic regulation during the T-cell differentiation. So thank you very much, Yi, for contributing to our seminar series, and we are very much looking forward to your seminar. Please go ahead.
All right. Thank you, Chuan. Thanks for the kind introduction and the opportunity to share my work. Today I'm going to talk about natural genetic variation reviews principles of epigenetic regulation in T-cell differentiation. So this work was done in the past few years in Rudensky lab at Memorial Sloan Kettering Cancer Center, together with Joris van der Weken, who is now a research group leader at Vienna, Austria. Wait, why I cannot go. All right. Okay, so first of all, T-cell is a type of immune cell. It migrates to periphery once matures in the thymus. Here is an overview of T-cell immune response. When the host is infected by pathogen, antigen-presenting cell activates T-cell, causing the T-cells undergo massive expansion to fight against the invader. After the pathogen got cleared, an activated T-cell will differentiate it into a population of memory cells that protect the host from the reinfection by the same pathogen. So on the other hand, regulatory T-cell repress this process to avoid harmful immune consequences and maintain immune homeostasis. So this figure describes a concept that the cell progenitor is like a ball on the top of the hill. While it rolls down, it becomes a terminally differentiated cell. We know that the ridge between the grooves maintain the cell fix once established. just like the transcription factors. Here, there exists many published paper demonstrate the chromatin remodeling is linked to the cell differentiation and identity establishment. And the chromatin remodeling is driven by the binding of transcription factor to the target cis elements, whereby gene expression is concisely regulated. Okay, take the CD8 T cell as an example. The engagement through MHC molecule and TCR during CD8 T cell activation is well known, regulated by a master transcription factor called TBAT, which is a T-box family member, of course. However, at the same time, there exists a panel of transcription factors we know expressed in the T cells. So we are interested in knowing how multiple transcription factors coordinate to bind to cis regulatory elements and orchestrate the chromatin remodeling to form specific epigenetic features, and how the formed epigenetic features subsequently affect the transcription factors regulatory behavior. So with all the underlying chromatin transcription factor dynamics, the T cell can then be reinforced to differentiate in the immune response process. So our goal is to dissect the overall transcriptional principles of T cell immune response. We have worked on these three key points in the immune response. For example, at activation state, we found that T cell activation is governed by a unique hierarchical regulation through multiple transcription factor coordination. At the memory state, we found T-box and the ROM families co-regulate the epigenetic and transcriptional memory in virus-specific CD8 T cell. While for the Tregs, FOXP3 shapes Treg cell identity and tunes the activity of transacting intermediates. So today I will focus on the T cell activation part. Although similar strategy was applied to all three stories. Okay, with the biological question I mentioned previously, so we utilize a hybrid mouse system here and set up a computational pipeline for deployed epigenome and transcriptome analysis. So we look into the allele-specific chromatin accessibility, gene expression, and multiple transcription factor families. The goal of the entire analysis work was aimed to figure out how the T cell activation is driven by different transcription factor families simultaneously. And what is the underlying transcriptional substructure specific to T cell activation? So the first question is, why hybrid mouse? So in 2011, Sanger Institute finished a mouse genome project where the genomes of a number of different mouse strains were sequenced. So take the cat strain as an example. So there are about 20 millions of genetic variation compared to Black 6 laboratory mouse strain. So if we cross them in the F1 offspring, there will be a large amount of genetic differences on the two alleles. This is like to perform like millions of genome editing at the same time. So if do the ATAC-seq analysis, you will observe at many polymorphism loci. Part of the sequencing reads come from Black 6 allele, and the rest are from the Cas allele. So as shown here in the black and the red bars. Again, our computational pipeline ensures the unbiased identification of allele-specific the source of the sequencing reads. Okay, so next, what is even more useful for the natural genetic variation is to identify the transcription factor binding motifs that globally affect chromatin accessibility. So specifically, when we see a genetic variation occurs in a motif and on one allele, if this motif is regulatorily active, we will observe the chromatin on the mutated allele become less accessible at this particular site. We know that almost all motifs exist throughout the genome thousands of times. We then ask, can we associate this genetic variation across the same motif with the corresponding chromatin accessibility and containing this given motif? So the answer is yes. If we see a globally association between genetic variation and accessibility, we can make a conclusion saying the transcription factor is actively binding to the chromatin through the motif in the particular cell site. In contrast, if we do not observe the global association, it indicates that the motif or the transcription factor does not directly bind to DNA or not important for this investigated cell type. This probably is a little bit technical. So here in this metric we use for X motif X, we separately select all ataxic peak containing motif X that is stronger either on B6 or Cas allele, and calculate the allelic ratio of peak in the two groups and sort them in a ascending order. If we see a separation of these two discrete curves, it indicates motif X is a active motif because variants in motif X systematically affect the chromatin accessibility on either allele. So conversely, in the case on the right side, motif Y is not an active motif because the genetic variants do not have effect on accessibility, as you can see here. Okay, here is experimental design. So for the T cell activation, we collect CD44 negative, CD62L positive splenic T cell as the naive cells from the hybrid mouse. We also infected the mouse with LCMV. 7 days after that, we collect tetramer positive CD8 and CD4 T cell as the activated ones. With these cells, we look the chromatin accessibility, both bulk and the single cell levels, and we also study the transcription factor binding profile for 8 different families using cut and run. Additionally, we look the distribution enrichment of all 4 chromatin remodelers as well as gene expression measured by ANSIC. Okay, and here on the left is the flow gates for sorting the target cell population for downstream study. On the right is it's the identified motif experiments that regulate the resting and the activated CD8 T cell epigenome here. In the first row, it's the BAF is stronger regulator for the virus specific CD8, because the delta mean, the gap between these two curve is larger. So, and while the left one motif from the SOX family significantly strong on the naive state, but as you can see here in the third row, the PAL2 seems to be a weak repressor for both naive and activated cells. So here I show you the BAF motif effect for naive and activated states separately. If plot the two delta means on the x and y axis, and we got a scatter plot showing all transcription factor motifs for naive and activated states in CD8 on the left and the CD4 on the right. As shown here, X, Runx, and SOX families are three most dominant motifs in the two cell type. Therefore, we decide to take the close look at them. Okay, we then performed the cut and run analysis assay for S1 and RUNX1 and the TCF1 to identify their binding sites across chromatin. So surprisingly, these three factors binds to up to 93% of all accessible genome, as shown in the Venn diagram. The gene chart here and show a accessible site is occupied by all three factors, and a genetic variation exists within the binding site, indicating a stronger binding on the Cas allele, as shown by the red bar here. Previously, we do the motif analysis using the allele-specific attack to identify the epigenetic remodeling transcription factors here on the bottom. By using the transcription factor binding data, not the attack data anymore. This is the transcription factor binding data from the cut and run. So we could identify to what extent transcription factor binding depends on its cognate motif. Here we see that S1, RUNX1, and the TCF1 indeed require their cognate motif for binding. And even more importantly, we could also know RUNX1 and TCF1 binding is strongly affected by S motif variation. Interestingly, chromatin accessibility of S1 bond site was to a large degree identical across naive and activated CD4 and CD8 T cells, as you can see in the scatter plot, the red dots. And when you compare these red dots, so the sites bound by RUNX1 and TCF1 in the absence of S1 underwent widespread activation-induced chromatin remodeling. More intuitively, if you look at the bar plot on the upper right, this was unexpected for us when we saw it in the first time. So it indicates that S1 binding sites have higher accessibility in naive state and maintain it in the activated state. So further categorize, we categorize the S1 bond sites showing a higher fraction of promoter cis elements. Accordingly, gene near S1 binding chromatin region were constitutively expressed at high level and enriched for gene involved in metabolic process. All right. Although S1 binding was largely similar between activated and naive T cells, our analysis revealed a small subset of sites that gained S1 binding upon T cell activation. So this is the red dots on the upper left corner. These elements were much less accessible in naive T cell, as you can see, and were nearest to the immune response-related genes with expression strongly induced upon T cell activation. So these upregulated S1 sites typically overlapped with intronic and distal regulatory elements that were almost always co-occupied by multiple activation-induced transcription factors, which I will talk in the next slide. So motif enrichment analysis showed that these X1 upsides were relatively depleted of intact X motif and match the canonical X motif to a less degree. As you can see, probably the small arrow point there. So, but this small subset are reached for the stronger B-zip and T-box family motif in the upper right corner, the green dots there. So this raises a possibility that S upsides, the small S upsides require activation-induced transcription factors to increase its accessibility during T-cell activation. Okay, so based on the motif analysis, so we pick 5 activation-induced transcription factor for deep study. So so they are TBET, c-Jun, and IRF4, BHLH, E40, and in fact C1. So their binding in activated CD8 T cell determined by common round indicate a range of 5,000 to 15,000 sites on the genome. Additionally, for each of the transcription factor, more than 50% of the peak co-bind by S1, as shown by the gene track on the top. If no S1 binds to the site, activation-induced factor co-localized with open chromatin region, whereas sites bound by S1, its accessibility does not present a big difference. Even the 5 activation factor induced factor binds there, as you can see. Okay, so this the bar plot here clearly demonstrate that most of this binding sites of these factors overlapped with S1 and underwent only minor changes in accessibility during T cell activation. So only those that did not over with S1 binding underwent dramatic activation-induced chromatin remodeling. So this heat map is another form of showing motif effect on the transcription factor binding data. On the x-axis are the motifs, and the y-axis represents each transcription factor. It tells us activation-induced factor needs their own coordinates and motif for binding, as expected, and X motif strongly assists the other 5 binding to their own target sites. So we further evaluate the combinatory effect of the activation-induced factor on the chromatin accessibility change at the S1 unbound sites. We found that the more factor binds, the higher accessibility change is induced. Okay, so therefore, our evidence suggests a simple model in which the functions of activation-induced transcription factor are overlaid onto the pre-existing chromatin landscape of naive T cell. As I already shown, the Zhong's motif contribute to chromatin accessibility in a large constitutive manner, and the same as H1. However, in contrast to H1, we found that the Zhong's 1 binding was detectable at nearly all accessible sites across the genome, including those gaining or losing accessibility upon T cell activation, as shown by the scatter plots. So this demonstrates upon T cell activation, Zhong's is like redistributed throughout the chromatin. So figure on the right at more information showing large fraction of up and down sides are enriched with Zhong's motif, whereas left one motif or SOX1 family motif is enriched in down sides, and back F and other basic family motifs are enriched in the up sides. Okay, this observation motivated us to see the correlation between RUNX1 and binding intensity and the chromatin accessibility change. So surprisingly, as you see from the scatter plot, surprisingly, these two measurements correlate extremely well with Pearson correlation equal to 0.8. The bar plot on the right shows the same correlation calculated for TCF1 and S1. So both of their values are less than the RUNX1. Interestingly, when we try to address if other motifs other than X family motif could affect RUNX1 binding, we do see BZIP, IRF, T-box, and the BHRH family motif assist RUNX binding. So based on this observation, we propose that RUNX motif, RUNX transcription factor may act as a regulatory signal amplifier that synergize with other transcription dependent activation dependent transcription factors. Okay, so the last transcription factor we studied here was TCF1. In contrast to RUNX1 binding to sites either gaining or losing accessibility, we found that TCF1 bonds preferentially to sites that lost accessibility as shown here. Again, this motif analysis using attack data I showed earlier indicates TCF1 positively associated with chromatin accessibility in naive T cells. So thus this cis element that bind by TCF1 but lost chromatin accessibility upon T cell activation is likely or very likely due to down expression of TCF1. So when look at the TCF1 binding intensity and reviewed by Cameron Run, we found the reduced binding upon activation was more pronounced in CD8 than in CD4. And when you compare these two scatter plot on top, so the left is CD8 and the right is the CD4. So you see the CD8 is turned angle to the naive CD8, which is on the x-axis. However, we found that there are sites that lost TCF1 binding by as much as like fourfold in activated CD8 still maintain their or increase their accessibility, suggesting loss of TCF1 is compensated by the recruitment of some other activating transcription factors. So indeed, this sites of losing TCF1 gained accessibility if one or more activation induced factor are recruited, as shown in this cumulative fraction of peaks. This is true for both CD8 and CD4 T cell. So this observation suggests TCF1 may frequently act as a placeholder in naive T cells for the later binding of activation-induced transcription factors. In addition, I also should mention that a small subset of TCF1 targets that acquire TCF1 binding only in activation state, as shown in green dots and on the upper left corner there. So we call the TCF1 upsides in comparison to the previously S1 upside. So similarly, a number of activation-induced factors boost TCF1 binding at this small peak group. The gene track is an example showing gain accessibility upon activation, and this particular side was bound by IRF4, C-Jun, NFAT-C1, and BHRHE4, but not TBAC. So we were curious to see if the TCF1 upsides overlap with the S1 upsides. So we calculate the overlapping cis elements, the number of the overlapping cis elements. It turns out these two peaks groups overlap not that much, as you can see in the Venn diagram there. So interestingly, we found that similar to S1 upsides, the TCF1 upsides were associated with immune response related genes, as well as gene involved in signal transduction, cell adhesion, and so on. So we saw TCF1 typically acted as a naive T cell specific activator. So our results suggest that it plays additional previously unknown role as a placeholder for activation-induced transcription factors and as a activation dependent cofactors. So the remaining question is whether the active motif or the transcription factors we identified using allele-specific taxic data also holds true for different T cell subpopulations, given the heterogeneous CD4 T cell population. So therefore, we performed the single cell taxic assay, and after data processing, integrating cell population markers, we obtain naive effector and the memory T cell subgroups, as shown on the right. We plot the single cell attack data by aggregating the cell type specific read counts. Therefore, present here as a pseudobug gene track. And as you can see that certain peaks highlighted in gray are accessible only in certain cell types. So consistent with our bug cell analysis, S1 bound sites showed very high chromatin accessibility across all T cell subsets. Whereas regions bound by the activation-induced transcription factors in the absence of S1 were dynamically regulated. So the S1 bound site is on the upper side, upper left. So the rest of 5 panels are the sites at S1 negative. Okay, so additionally, a comparison between the two main subpopulations of CD4 T cell arising in response to viral infection, Th1 cells and the TFH cells, revealed a stronger effect of the T-box motif variation on the chromatin accessibility in the former, and a stronger effect of SOX motif variants in the later. This effect was conserved mostly at regions not bound by S1, as you can compare on the two scatter plot here. Okay, so I briefly summary. So first, we built a hybrid mouse model, plus a deployed epigenome analysis pipeline, identifies the active transcription factor binding motif. S1 binding defines constitutively accessible chromatin. RUNX1 can amplify genome-wide chromatin accessibility changes, while TCF1 acts as a placeholder for the activation-induced transcription factors. And the last, immune response genes recruits S1, a small subset of S1, and the TCF at a small cis element size upon T cell activation. Okay, and that's all my work here. And so thanks my postdoc supervisor Alexander Rudensky and Joris van Weken. So they are great guys to work with. So here I'm also do a advertisement for my own lab here in Shanghai Zhong University. Sorry. Okay, so that's all my work. Yeah. I'm happy to take any questions.